Mine, yes, mine's on.
Oh, thanks. Okay, I think we'll get started since it's close to 7:30. So I think Troy needs probably very little introduction. He's been here for quite a while at the University of Minnesota, but I'll briefly tell you about him. So he received his bachelor's here at the University of Minnesota, and then left us for a short while to get his master's and PhD at the University of South Florida, and then came back here for medical school, residency, and fellowship. And he's been on faculty here since 2009. And he's currently an associate professor. So his areas of research are multiple, the main areas being hematopoiesis and stem cell homing, as well as metabolic disorders, including cerebral adrenoleukodystrophy and Hurler syndrome. And he also holds an appointment in the Division of Global Pediatrics and goes to Africa relatively frequently to work on hematologic disorders there. So today, Troy's going to talk with us about stem cells.
All right, thanks. So I'm going to tell you a story about that we've been working on for, I don't know, maybe 5, 6, 7 years now about stem cell trafficking. It's a little part of the lab, but the lab is all combined together. There's kind of one general theme, and it is all about the transplant process in any of the diseases that I study. And so we study how the stem cells move around the body, whether the stem cells are going to the bone marrow, or whether they're going to the brain, or whether they're remodeling some other organ. So let's start with a brief introduction, and the talk is kind of aimed at learners in the audience to understand the basics of stem cell transplant. And why do we do it? Well, it's no secret that it can be curative for some malignancies, leukemias, lymphomas. We use it in non-malignant diseases like sickle cell disease, thalassemia, immune deficiency, etc. And then in this unique realm of storage diseases, bone marrow transplants can provide missing enzymes in the diseases such as mucopolysaccharidosis type 1, adrenoleukodystrophy, beta-chromatic leukodystrophy, etc. So bone marrow transplant was pioneered here at the University of Minnesota in August of 1968 is when the paper was published, so it's just over 50 years now. My PhD mentor, Robert Good, is shown here on the left. He was the leading physician performing that first transplant. It was performed across the river, not far from the old hospital, actually. And fundamentally, it's pretty easy. Bone marrow from one person is infused into another. Back historically, these transplants were all based on bone marrow, but then through the years, alternative stem cell sources were developed. Umbilical cord blood became a stem cell source, and you can also mobilize bone marrow cells into the peripheral circulation, harvest them, and use them as a transplant source. So this was, in fact, the first stem cell therapy. Even though it's a very popular phrase these days, we've been in practice now over 50 years with it. So there are risks, of course, going through transplant, and this ties into my project. The use of ablative chemotherapy increases your risk of infection during transplant, especially because you wipe out the immune system and then you have no neutrophils and you're subject to increased risk of infection. Recently, when we've tried to get away with using less chemotherapy, we call that non-ablative transplant, the problem is the graft doesn't take and you experience graft failure. So we're always constantly looking for that magic balance between how much chemotherapy is too much and not enough. And you can have this entity of graft versus host disease, which is an immune attack against various organs in the body, skin, liver, gut, etc. And that has the highest risk with non-sibling donors and those who are not HLA matched. So let's think about engraftment. So the y-axis looks at is looking at the incidence of neutrophil recovery after transplant. The x-axis is days after transplant, and you recovered when you have about 500 neutrophils per microliter for 3 days. That's what we term recovery. The process of recovery after a transplant often takes about 28 days or so. You can see that the majority of the patients in this study are recovered, looking at the black line here. It's during this period that we see the highest rates of infection and the highest rates of relapse. And so we're constantly trying to push this curve to the left to increase the speed at which neutrophils are recovered. So if we can decrease the time of neutropenia, perhaps patients will have less infections, they'll have less time in the hospital, and less overall mortality. So the story that I'm going to tell you, it always starts with conditioning, and we use this term often on the wards. What is conditioning? Fundamentally, conditioning is just either chemotherapy or radiation that we give to our patients to prepare them for the graft. Historically, radiation has worked very well. We use total body irradiation, 1360 gray. And then in the modern era, busulfan is a very powerful alkylator, which probably works equally as well as radiation. We often nickname this radiation in a bottle. Perhaps there are some different types of side effects that occur with busulfan that you don't see with radiation and vice versa. So fundamentally, the field has moved away from radiation towards just using busulfan, but they're equally potent for engraftment. This preparative regimen, or the conditioning, does three things. It removes residual cancer cells. It makes space in the bone marrow for the graft. So if you have sickle cell disease, you want to get rid of all the cells in your bone marrow so you can accept the new graft. It removes T cells that could lead to rejection of the graft. And what's important for my lab is that it establishes the signals needed for new stem cells to migrate into the bone marrow. We call that process homing, and you'll hear me use that term a lot. So it sets up the signals so the cells can home and engraft into the marrow. So again, going back to the fundamentals, bone marrow is taken from the donor and infused into the recipient. Again, we have alternative sources like cord blood, we have peripheral blood. And for people who have not seen a transplant, it literally is probably one of the most boring procedures in the world. It literally is a bag of product, of this bone marrow, that's hooked up to an IV and goes into somebody's arm. There's no surgery. A lot of questions from the families are, when is the surgery? When am I getting the marrow implanted? It doesn't work that way. You can actually squirt marrow into somebody's bones. It's very difficult because of the pressure differential. But in my review of the literature, in my opinion, there's no benefit to doing that. So we just infuse it right into the arm. And then what happens after that is really what we try to study in the lab. So after that is the homing process. So somehow all those bone marrow cells go from the bag through your arm, through the organs in your body, and they end up in your bones over the next 30 days. And what is commonly thought is that you have one shot at that. So this homing process takes place probably within 24 hours. And if your cells haven't homed to your marrow in 24 hours, it's a done deal. You're not going to engraft. And so understanding this process becomes fundamentally very important. So it's like running a race or a hurdle race. So getting those cells from your arm to your bone is like these hurdlers. Some of the cells do very well, are just flying into the bone marrow. Some of them get caught, some of them move a little slower, some are just destroyed, and some just give up. They don't make it. And it's probably somewhere on the order of about maybe 10% of what's in that bag actually makes it to your marrow. The rest of the cells, we don't know their fate. They just disappear. Either they undergo apoptosis, they get caught in the lungs, they get caught in other organs. We don't know. But if we could speed up this process, again, we could move that neutrophil recovery curve to the left and speed up engraftment. So going back to conditioning, how does this whole process work? So as I mentioned, we give people radiation, or now busulfan, and that clears the marrow of residual host cells. It also sets up an incredible amount of inflammation because it's an injury. And you get a release of all sorts of inflammatory factors, and these are just small molecules and proteins that your bone marrow starts releasing after it sees busulfan irradiation. And these serve as the signals for hematopoietic stem cells to migrate inwards. There have been a lot of labs and a lot of attempts to understand what are these factors that are encouraging stem cells to move into the marrow. And if we could figure out those factors, we could actually turn them into drugs or to accelerate engraftment, either through enhancing their effect or actually making the factor in a lab and giving it to people as extra homing factor. So like I said, stem cells respond to these factors and they're drawn into the marrow space. So after radiation or busulfan, you end up with this gradient of large amounts of these proteins in your bone marrow that go to lower amounts in the peripheral circulation, and the stem cells that are infused in your arm actually migrate along this factor gradient. So they go from the low concentration, and they enjoy these factors so much, they want to reach to the source of them, which is the bone marrow. And that's fundamentally how we think it all works. And so this is a nice pictograph of the process. This is a vessel running through the center of your bone. These are infused stem cells circulating through the vessel. And these are the endothelial cells that make up the vessel on either side. And so there's all sorts of different receptors and interplay between the stem cells here and the endothelial cells. But eventually, the stem cells squeeze between the endothelial cells and they get to this area here, which is the bone marrow proper. We call that the niche. And all those factors I've been discussing are released from the niche, and they're probably released from the endothelial cells, and that's why the concentrations are so high in the marrow. And then they diffuse out into the circulation. And bone marrow stem cells really enjoy these factors, and so they migrate towards them and end up here, and this is successful homing and engraftment. So back to conditioning and the premise for the hypothesis of the talk. So exposure to busulfan irradiation is a very inflammatory process. It's also not normal. Our bodies were never meant to experience busulfan, certainly, nor were they meant to experience 1360 of radiation. So it triggers a lot of injury. And again, that injury is what is key to the whole process because that makes us very inflamed. Most responses in the body are counterbalanced with an opposite effect. The examples are insulin regulation, bone marrow or bone remodeling is driven by osteoblast, osteoclast balance. Wound healing is driven by a scar buildup, scar breakdown. And of course, classically, bleeding and clotting have two sides to the coin. So we believe there are factors that promote homing. These are very well studied, actually. I can name off 6 or 7 of them: SDF1, LAMP2, and a whole bunch of others that promote homing. There are also probably factors that inhibit homing. This is completely unstudied, and this is where the story for the lab begins. So we hypothesize there are factors that actually inhibit stem cell homing and engraftment after transplant. Such factors serve as hurdles to the transplant process. Remember that hurdle picture? It all kind of ties together. So, and knowing the factor's identity allows us to target them and potentially reduce the hurdles, thereby speeding up transplant. And so classically, we would phrase this in a null hypothesis, that there are not factors that inhibit hematopoietic stem and progenitor homing, or HSPC homing, and our job is to dismiss the null hypothesis. So this is what we've been studying, is what are these factors that might inhibit stem cell homing? Do they even exist? So how would one go and answer this question? Well, a few years ago, and probably 6, 7 years ago, we actually introduced zebrafish into the lab to work on some of these questions. And so we thought, well, why don't we just go and look for genes that are changing in response to radiation in the niche? And we decided to start in the zebrafish. And why did we do this in the fish? Well, we can actually isolate the bone marrow niche in the fish easier because their niche is kept in the middle of their body and it's not mixed in with their bones. isolating cells mixed in with bone structure is exceedingly difficult because bone is a calcified tissue and it's hard to actually remove the cells. The genes in the fish and mammals and people are nearly the same that control bone marrow transplant and control engraftment and control hematopoiesis. The zebrafish are established as a transplant model. This was done by the Zahn lab about 15 years ago. We adopted it into our lab. You can actually perform many bone marrow transplants using donor and recipient fish. The recipient fish receive radiation just like a person would. You can actually give them busulfan, and you can infuse bone marrow into them, and they'll engraft over a period of about a week. So it's a pretty good model for this question. So the first problem we encountered was how do we separate the stem cells, the hematopoietic stem cells, from those niche cells? Because what we're really interested in are the niche cells, the cells that are making all the factors. Well, there are two facts that helped us. Stem cells are very small, niche cells are very big, and we had a filter in the lab. And so we could actually put the marrow of the fish through the filter and separate the niche cells from the hematopoietic stem cells, because we didn't want to study the hematopoietic stem cells after radiation, because that would skew all the results. So we just wanted to isolate the niche cells, and this is actually a photomicrograph of zebrafish marrow. The stem cells, the hematopoietic cells, are the little dark purple ones here, and then everything else in it is a niche cell. So we want to get rid of these guys, and we just did this through a filter process. And so we tested, so the setup was you radiate the animal, you isolate the niche cells, you prepare RNA, and then you plot the data. We tested the system with some pilot experiments looking at the RNA levels for hematopoietic genes versus niche genes early on. And we looked at the differential between the cells in the top of the filter and the bottom of the filter. Again, the bottom of the filter is going to have the small hematopoietic cells. The top of the filter is where the big niche cells is. And so when you look at the differential between the RNA between niche genes and hematopoietic genes, you actually see that the filter process works quite well. These genes on the right are all expressed in niche cells, and we found them very highly upregulated in the cells in the top of the filter, and then all the small cells that flowed through the filter expressed all these hematopoietic genes in red. So this proved that the system actually worked pretty well, and it was just a simple filtering technique that we used to separate these cells. And then we did the same process again, but we took the niche cells that were on top of the filter and we put them on a gene chip. So we looked at every gene in the zebrafish to see which genes are in fact upregulated by radiation. And you get plots like this. You get the green genes that are downregulated with radiation, and you get the red genes that are upregulated in radiation. You can see there's a couple hundred on either side, and then you get probably 28,000 genes that aren't changing very much. So it's very few genes that change with radiation, and we know the names of each of these genes. And so we basically looked at the list, which looks like this. We looked at the upregulation, and then we did put some thought into the list. We didn't just pick one gene. We were really looking at things that were secreted or bound to extracellular matrix or things that were studyable. We didn't want to study nuclear genes, even though they were upregulated in response to radiation. We didn't want to study cytoplasmic genes. They were less interesting. We really wanted to study extracellular genes, again, things that are secreted. And so at the top of the list, we find a gene called dermatopoietin, and that's where we've focused a lot of our attention in the last 5 years. So dermatopoietin, well, abbreviated DPT, is what we chose to focus on. And first we verified that dermatopoietin is a gene that's increased in response to radiation by looking at RNA levels. We actually did immunohistochemistry for dermatopoietin in the zebrafish marrow, and this is shown in red here. So after radiation, we can see that there's a lot of dermatopoietin increase in its protein level. And that kind of ends the zebrafish part of it. There were a lot of other experiments that didn't necessarily work, but in the zebrafish, what we were able to show is that dermatopoietin which turned out to be a relatively unstudied non-collagenous protein. It's 23 kilodaltons, so it's a little bit smaller than the typical protein. It is associated with the extracellular matrix, and it's really found in lots of organs and tissues, from tendons and brain and intestine and lungs. It's also found in the circulation, so we all have it. It circulates through us. And most importantly is you can buy the stuff. And sometimes that's as important in the lab as anything else, is that you want to have a reagent you can work with and do experiments on. So if companies sell recombinant dermatopontin, that makes your life a whole lot easier because you can use it in experiments. So I say it's relatively unstudied. So when we first started working on this, there were maybe 40 papers in the world on dermatopontin. It was very surprising to me. It really went unnoticed by many labs. The papers that were out there were a whole variety of different descriptions, and the papers actually weren't very good. They're pretty low level. But some people theorize dermatopontin is involved in clotting. That is, it makes clots harder or better knit together or tighter. It may be involved in wound healing. High levels are found in scar tissue, which I'm not sure what that means, but that's what one paper showed. Another paper showed it's involved in cell adhesion, at least in the lab when we test this in plastic dishes. It has been thought of as a marker of a metastatic disease. That is, cancer cells seem to have higher levels of dermatopontin for an unknown reason. It perhaps aids in skin integrity. It is thought to perhaps be, if mutated, be a form of Ehlers-Danlos syndrome, though that's never been conclusively shown. So this variety of mishmash of papers seemed to be there was like an adhesion theme here going, but we weren't really sure. So I guess we do have two more key zebrafish experiments. One is a bone marrow transplant using, in this case, we use these green glowfish. We use bone marrow cells from these fish because they're really, really bright. You can see them under the microscope, and you can actually transplant the green fluorescent cells into a non-fluorescent fish and mark the engraftment, and actually you can count how many cells make it to the bone marrow in real time. So we transplanted a bunch of fish with green bone marrow cells, and 24 hours later, we counted how many cells made it back to the marrow. And we did this in the presence of dermatopontin, again, which we ordered, and the absence. And what we saw was this, is that in the control situation, we had about 100 positive cells per 100,000 marrow cells, and dermatopontin, when we mixed in this protein with the donor product, we lost about half of them. So this was the first evidence that we had in the lab that dermatopontin, if it did anything, it actually inhibited the entire process. We then answered the same question with another approach. We used a fish we built in the lab in which the bone marrow cells express white light. And you can do the same transplant process. You can take these cells as donor cells, transplant them into a fish that has none of these special cells. The benefit of doing this is that when you want to look at homing and engraftment in the recipient fish, all you have to do is put the fish to sleep and image them under a special camera. You can actually quantify the numbers of cells without destroying the animal, which means you can do this every day for a period of time. And so this is an example of two fish together. The bone marrow of the fish, or the marrow of the fish, lies right here behind the heart and then extends downward towards the tail. And again, this is the same experiment where marrow cells are mixed with albumin or a control, and you get this nice amount of bone marrow homing in 24 hours. But when we mix the cells with dermatopontin or DPT, we see none of that. The cells are just lost. They get stuck somewhere or they undergo apoptosis. So again, now this is the second piece of evidence that dermatopontin, in fact, reduces stem cell homing, in this case, probably by up to 60% or so. So what we've learned from the fish experiments is that dermatopontin It's increased in the marrow after conditioning in the zebrafish, and it inhibits cell homing to the marrow in the zebrafish. So what do we do next? Well, we next move into our favorite animal, which is the mouse. It's more classically studied. There's a lot more reagents to use. Fundamentally, it's a bit closer to humans. And so we test the entire system now in mice. And so the first thing we had to measure was, is dermatopoietin in mice, does it go up after radiation like we saw in the fish? And so we did Western blots to look at the amount of dermatopoietin in circulation after radiation, and that is shown in here. So this is just plasma run prior to radiation, then 1, 2, and 3 days. And looking at dermatopoietin, you can see there actually is a spike at day 1 and day 2, and it goes to baseline at day 3. We can measure the signal here by scanning this gel. And we did another type of assay called an ELISA, which uses antibodies to pick up dermatopoietin, and we see very similar results. That in fact goes up in the circulation after radiation and then returns to baseline by 3 days. So it recapitulates our initial data in the fish. So then comes this homing type of experiment, just like the fish. We set it up like this. We give mice radiation. We give them dermatopoietin 24 hours later in their tail vein. And then 30 minutes after that, we inject the other side of the tail with the transplant. And then we wait for 24 hours or so. And then we look at how many cells went to the bone marrow. And for this type of experiment, we have to sacrifice the mouse and take out the bones and count the cells. Oh, and I should emphasize that what happens to circulating dermatopoietin when we inject it like this? And so we did another Western blot, and we can actually measure what we injected is called recombinant dermatopoietin, as shown here. This is pre-injection. This is 1 minute post, 30 minutes post, 60 minutes post. So the dermatopoietin actually has a half-life, and it's probably around 20 minutes or so. So that's why we perform this transplant within 30 minutes, because we want to capitalize on our injection. And so then we look and count the number of stem cells that have homed to the marrow the next day. And just like we saw in the fish, in the control situation, we have about 18,000 cells homed to the marrow. But in the presence of high amounts of dermatopoietin, that homing process is completely shut off. We've inhibited probably 90% of the cells reaching the marrow. So this is our first indication that now in a mammalian system, the protein is doing something strange. It is causing stem cells to not make it to the marrow. They're getting bound up, or they're dying, or something's happening. There's another type of assay for stem cells. When you look at the history of bone marrow stem cells, there are thousands and thousands of papers and lots of definitions and lots of experiments you can do. Another surrogate assay for a hematopoietic stem cell is called a spleen colony forming unit assay, and it's very old. What turns out to happen is that when you give an animal a graft, some young stem cells migrate into the spleen, and they actually are white. And these cells grow and divide, and they make a colony in the spleen. And at the right time, you can actually take out the spleen and count these colonies. We call them spleen colony forming unit cell assays. And this probably happens in people too, as the spleen starts off and engrafts and repopulates with the donor cells. We're not sure why they're white, but they make the experiment work because you can count them. And again, they're a surrogate for stem cells. And so we count these colonies 8 days after transplant to give us an idea about stem cell homing to the spleen. And when we did this in mice, we have about 40 colonies or so per spleen per mouse. And when we did this in the presence of high levels of dermatopoietin, you can see we cut that value in half. So now we have additional evidence that hematopoietic stem cells are being inhibited. So then there's another type of an experiment. It's a long-term transplant experiment. So these are all very early readouts. So within 24 hours of infusion, within 8 days of infusion for the colony-forming unit assay. What you really want to know is, does this translate to reduced engraftment many months and weeks later? And so we did the type of experiment very similar, where we give dermatopoietin, and then within 30 minutes we give the transplant. But now we wait. We wait and assess the animals at 1 month, 2 months, 3 months, and out to 6 months, and we look at the amount of engraftment they show. I should emphasize that in this case, we didn't use ablative radiation, we used low radiation, and that's very important. Because if you give a mouse totally radiation to wipe out all its bone marrow, and in this case it's 11 gray, and you inhibit the product, what happens? The mouse doesn't engraft and it dies, and you have nothing to measure, because you can't measure a dead mouse. Any mouse that survives totally radiation has 100% engraftment. What we wanted is an admixture of immune systems because we can count it, we can enumerate it. So we give them low dose radiation, and that allows almost a perfect partial engraftment. And I'll show you what that looks like. So that is shown in the blue dots here. So we measure the donor engraftment in a mouse that receives partial radiation, in this case 4 gray, and we measure it in the peripheral blood, and we see that engraftment's rated about 60%. It kind of wobbles between 50 and 60% in a controlled condition without any permutation. When we give dermatopontin within the first 30 minutes of transplant, you can see the amount of donor engraftment we see is significantly lower. We're at 20 to 30% levels in our model system here. And these data points are all significantly different. And we measured this engraftment at 21, 60, 100 days, 3 months. We've actually measured it out to 6 months, and the data holds perfectly. So what this means to us is that this initial setback in homing turns out to make the transplant less efficient in engraftment and does affect long-term engraftment as well. So with this part, we've determined that high levels of dermatopontin inhibit hematopoietic stem and progenitor cell homing. That's kind of our common term now for hematopoietic stem cells, the short answer, HSPC. High levels of dermatopontin inhibit CFU spleen assays, as I showed you, and also high levels of dermatopontin inhibit long-term engraftment. So those are all the pieces of data we put together. So now what? Well, scientifically, what we've been able to do is take a system, and again, you should remember that the mice have their own dermatopontin in the system. And all we did was augment it way up by injecting dermatopontin we bought from a company. Wouldn't it be great if we could knock out dermatopontin and then measure all these different things? And if we saw changes in engraftment, that would really be conclusive. And what we would predict is if we delete dermatopontin from the mouse, engraftment should speed up. So one can actually engineer animals pretty easily these days. You can almost buy animals by mail order, and you can engineer them with any gene deleted. And so we made a mouse in the lab that has dermatopontin knocked out or deleted. And so that was the next step. So our statement was, we rationalized the best way to prove that GPT is an inhibitor is to genetically remove it from the mice and then test engraftment. I'm not going to go through how we did all this. We used modern technology. It's CRISPR-Cas9. It's a series of templates and enzymes that you can inject into the nucleus of basically a zygote, a mouse zygote, and it'll actually knock the gene out. You can get both copies of the gene knocked out at one time. It can be very efficient. You grow up those mice, you cross them out, you cross them in. Eventually you get a mouse that has both the dermatopontin genes completely gone, you verify that, etc. What was important for us is that sometimes you knock out a gene and it's necessary for life. In this case, dermatopontin was not. The mice are actually viable. Sometimes you knock out a gene and the mice can't reproduce, and that really puts a stop to things. But in this case, the mice actually still reproduce, so that's great. And we saw no obvious changes to peripheral white counts, platelets, things like that. There might be some subtle changes, but nothing obvious. And the mice weren't dysmorphic, so they didn't have funny shapes. The bone structure was pretty normal. One thing that is interesting about the mice is that their skin is kind of lax, actually. You can pull on their skin. It just seems to pull away a lot easier than a wild-type mouse. This is completely unstudied in my lab because we're not skin experts, but it's something that we noticed. And it's pretty consistent. So this is the structure of the gene. This is exon 1. We've knocked it out. We've inserted a bunch of stop codons. This is proof that we knocked it out. So looking at the plasma for dermatopotin, we have the heterozygous mouse, we have a wild-type mouse. In the middle is the knockout mouse, lacks any dermatopotin. This is the peripheral white cell counts of a wild-type mouse. The red is the knockout mouse. There's no difference between the blue and the red. In this experiment, the heterozygous mice had a somewhat increased white cell count. I can't explain that. We've never really figured it out. But it showed we could do this. So getting at the first fundamental experiment, we just did everything again. We did this homing experiment where we gave mice radiation, we injected them with a recipient bone marrow, and then we looked at the numbers of cells that were in the marrow 24 hours later. And we do this in wild-type mice, you get about 20,000 cells, and in our knockout mouse, it turns out you get about 30,000 cells. So that's an increase of home cells by about 30%. And that data is statistically significant, and this represents probably about 15 animals per group. So it's the first evidence that in this knockout mouse that, in fact, homing does speed up. You can do that CFU spleen assay I talked about. This is the prior data, the wild type, and then the recombinant dermatopotin condition. Now we did the same thing in the DPT knockout mice, and it looks like we've increased spleen forming units by about 5-fold. So they go from about 40 up to about 100 and 120, 130. So a tremendous difference in this colony spleen assay. Same thing we did before, we can look at long-term engraftment. We did this with low-dose radiation, so we would have partial engraftment. That way we would have something to measure. And so these are the wild-type recipient mice here. They engrafted at about 40% from 5 weeks to 20 weeks, and we pushed that all the way out to 6 months. And in the DPT knockout mice, again, looking at how they engraft, turns out they engraft about 30% better, so they engraft at about 60%, and that is maintained and consistent throughout time. You can actually do transplants from these mice into additional mice, and that advantage to engraftment actually persists. So what it tells us is that without dermatopoietin around to inhibit things, stem cells actually get to the marrow faster, and when they get to the marrow faster, they affect the entire long-term engraftment process. There are very particular ways to characterize hematopoietic stem cells by cell surface markers. When you do that on flow cytometry, we put names on the cells like LSK and LSK SLAM. These represent probably the truest of true stem cells. We can actually count those cells in the marrow of a mouse. And we did count those cells to really assess fundamentally, are stem cells affected? When we transplant dermatopoietin knockout mice and wild-type mice, it turns out yes, the DPT knockout mice have a lot more of the recipient stem cells. When we look at LSK and LSK SLAM, in either case, there's more of them there. So now with the dermatopoietin or DPT knockout mice, what have we shown? We've shown that HSPC or stem cell migration and homing is faster, it's more efficient, and this translates to increased engraftment. And what this means to us is that when you take away dermatopoietin, everything gets faster and better. So now as a scientist in lab, now what do you do? So I think we have conclusive proof that this is an important protein. You always want to know, well, how does the whole thing work? Like we've been injecting dermatopoietin, we knocked it out, we didn't even know what it did. But now it'd be nice to know, what does it do? So how is it inhibiting this process? So let's go back to the first step after you infuse stem cells. Stem cells are going through the blood vessels and they're migrating through the vascular wall and they're getting into the bone marrow. And so we call this breaching the wall. So we can actually buy dermatopoietin, and in this case we bought some and we labeled it fluorescently green, and we put it on endothelial cells to see if it would stick. And it turns out that when you look at endothelial cells, dermatopoietin binds directly to the cells. When you actually infuse a mouse with dermatopoietin and then go back and look to see where the dermatopoietin is, This is the center of an aorta. And you can see using immunohistochemistry that generates a brown signal for DPT is binding to the inner surface here of the aorta. It's not in the control situation. So it looks like in initial experiments, DPT binds to endothelial cells and the intervascular wall right here. You can set up various chamber-type assays in which you coat specially designed wells that have holes in them with endothelial cells to make an artificial vascular wall. And this would be akin to the niche space. This would be akin to the circulation. You can then load these wells with hematopoietic cells. And over a period of 24 hours, the cells will migrate through the endothelial cells, through the holes, and you can count them in the bottom of the well. We call this a migration assay. You can mix in DPT and see what happens. And when we do that in the control situation without DPT, we get so many cells in the bottom of the well. When you add DPT, you actually reduce that cell migration by about 30%. So DPT is causing some poor interaction to occur between the endothelial cells and the hematopoietic cells here. There's another type of assay called an adhesion assay, where the endothelial cells are in the bottom of a well, and you put marrow cells on top of them, and you incubate them for 24 hours, and you wash them off, and you literally just count how many cells are stuck there. We call that an adhesion assay, and you can do that in a control situation. You get what we call 100% adhesion. And then when you add in dermatopontin and measure the amount of cells stuck, you actually reduce that by about 15%. So it looks like dermatopontin is fundamentally having an effect on the migration and adhesion of the stem cells as they go through the vascular wall. At least that's what our model systems are showing us. Through some collaborations in the biomedical engineering department, we've actually been able to build an artificial blood vessel on a microscope slide. It's a plastic barrier with slits in it, as shown here. You then can put endothelial cells down on the barrier, which coat the entire barrier and make an artificial blood vessel. I think I have a better picture here. And so this is all on a microscope slide, and this is the artificial blood vessel with coated endothelial cells. And again, there are slits in this, so you can actually measure the amount of cells binding and interacting with the endothelium. And so what's nice is that you can build this blood vessel on a microscope slide, and this is a hollow lumen, so you can actually infuse bone marrow cells through the blood vessel. And in real time, you can actually count and watch the interactions between the infused bone marrow cells and the endothelial wall. And so we call it vessel on a chip. And so we performed an experiment like that in the presence and absence of dermatopontin, and we looked in this type of assay what happens to the cells. In this case, we just looked at the amount of cells, the amount of bone marrow cells that were adhering to the vessel wall. This is the control situation. We get so many cells adhering. You can actually give TNF-alpha, and that enhances the wall stickiness, and we see a lot more marrow cells adhering to the vessel wall. When we add TNF plus DPT into the situation, we actually cut that adherence in half, so reduce it by 50%. So now I've shown you by 3 different ways how dermatopontin, we think, affects hematopoietic stem cells. We think it's right at this point. And we hadn't learned this before. It could have been doing anything like inducing apoptosis of the cells or cell death, but it looks like it's actually inhibiting some interaction right there. So the conclusion from the last 4 slides is that DPT interferes with HSPC adhesion to the vascular wall, and it somehow blocks transendothelial passage of cells. That's what we mean by breaching the wall. And so we have other data I'm not going to show, is that we've figured out which protein DPT blocks. We believe it's probably VCAM1 on endothelial cells, and DPT likely comes in, sits on that receptor, which is needed for stem cell homing, and blocks the marrow cells from reaching the receptor. And that's specifically how it's interacting. We have more experiments to do, but it's probably VChem1. So the final experiment is, okay, we've described elegantly how this all works, the discovery, the fish, the mice, everything. Can we do what we set out to do? Can we reduce DPT somehow? Well, as luck has it, it's a small protein and it has various domains, but I'll show you one slide, is that fortunately for us, it has something called a heparin binding domain, and it's a sequence of amino acids that binds to heparin. You notice in this case, we use the term heparan. It's related to heparin. They're essentially interchangeable, and heparin is the classic sugar that you all know about. It allows you not to clot, basically. It's a repeating unit of various sugars, and in this case, DPT has a domain which maybe binds to heparin. Despite you giving all your patients various amounts of heparin so they don't clot, we always have circulating heparin through our bodies to some extent as well. So I have experiments I'm not going to show you, but we actually showed that in a controlled situation, DPT actually binds to heparin, and that's a real interaction. It's not theoretical. So let's go back and we'll redo the experiment that we did originally to look at homing. We can give mice DPT, and then we can give them DPT plus heparin, and we can look at the amount of cells homed in the marrow. And what happens when you do that? Well, in the control situation, you get about 7,500 cells homed to the marrow. In the presence of all that extra DPT, you actually reduce that by 90%. But if you mix the DPT with heparin beforehand, you completely overcome the blockade that you initiated with DPT, and actually you get some enhanced homing. So now we've come full circle, and we actually came up with a way to inhibit DPT. We believe we don't know exactly what the heparin is doing, whether it's making the DPT completely non-functional, or whether it's breaking down to the DPT. We don't know that yet. I suspect it's making it non-functional, but we can completely reverse the process. So in summary, through this last 7-8 years saga, is that we've described stem cell homing process. It's monitored by things that enhance homing, and now we're one of the first labs that have discovered a protein that actually inhibits the homing process. We call that positive and negative forces. We've described DPT as a small extracellular matrix-related protein, and it fundamentally plays a role as a negative force. We've determined that DPT blocks efficient HSC binding to the endothelial cells, so it prevents cells from breaching the wall and reduces their entry into the bone marrow niche proper. We've shown the absence of DPT improves homing and improves engraftment, and now finally we have some pilot data that when we inhibit DPT, and at least in the experiment I showed you, we used heparin, that's predicted to produce an enhanced effect, and the pilot data shows that that is true. So back to our hurdles. So you don't want the guys stumbling and falling over the hurdles back here. What you want to do is you want to give all your stem cells a fair shot and allow them to get over the hurdles and reach the finish line, in this case the bone marrow, all at the same time, as fast as efficiently as possible. So this work has been undertaken by many generations in the lab. The first generation is shown in blue at the top with Tiffany and Michael and Bo. Second generation, Ashley and both of the Amandas contributed to it. The third or fourth generation is sitting up here in the fourth row. Willa, Justin, Brooke, and Layla. We have numerous collaborators around the country that have helped unravel this, and support from multiple sources that help make all the work possible. Thanks. That's the next talk.
With your 400 gray experiment, did you get the same increase in DPT after that So is there a gradient to DPT increase if you change the radiation dose or if you use busulfan?
So that's a really good question. We measured at 9 gray, which is ablative dosing, which is total ablation. We also measured at 7. We did see an increase at 4 gray, but not as high as 9. So there is an increase, but it's not as robust. And then busulfan, we did busulfan, it's exactly the same. Busulfan causes this giant increase in
myeloablative dosing, similar.
Yeah.
Did you also look at delaying stem cell infusion? So in a practical sense, could we add more rest days and avoid the increase in DPT and get better engraftment?
Yeah, that's an experiment I've always wanted to do. But in fact, some of the older labs around the country, Peter Quisenberry out east, did this experiment in the 1980s. They actually let mice go for a while, like 1, 2, 3, 4 days. And it turns out if you do that, the mice did engraft better. It was 20 years prior to dermatopontin, but fundamentally the answer is probably yes.
So, Troy, that was really beautiful and very impressive. So, obviously, in a transplant situation, you want the cells to breach that barrier, but that barrier must be there for a reason. So, my question is, are you worried about bad things coming in?
Yes. Well, so I didn't go into the teleologic explanation for this, but it goes back to we are not meant for radiation at 1,000 gray. We're not meant for busulfan. We think it's the injury. And so that's now a project in the lab that Justin has been working on is what is the regular role of dermatopontin? It's in fish, it's in zebrafish, it's in mollusks. It's highly conserved. What we think it is involved in endothelial repair. And we think it actually helps you heal after you damaged yourself somehow, and it helps your blood vessels heal. And so if you injure endothelial cells in a dish and add dermatopontin back into that injury, it doesn't matter how you injure them. It could be toxins, radiation, physical. The cells actually function better with dermatopontin around. They come in closer, they knit back together, and the collagen becomes tighter. So we think it's a repair mechanism is why it's there. We can overcome it in a transplant by giving somebody enough radiation and poison, but that's probably why it exists, is to repair your blood vessels.
Great talk, Troy. Very interesting protein. You mentioned earlier in the talk about something about Ehlers-Danlos syndrome, and are there human diseases characterized by polymorphisms in the coding content of this gene?
Yeah, so we were interested in that a while ago because we made this mouse separately from anybody else. There was a mouse 15 years ago made in Japan, same mouse, one single paper on it. The mice don't exist. We couldn't get them. They proposed making it to study Ehlers-Danlos syndrome. And they said, these mice have lax skin. This could be a great model. So then I actually contacted societies and foundations and kind of looked, has anyone ever described a mutation? And it turns out I can't find anything. There are a lot of undiagnosed patients out there still. So it could still be true, but I haven't found a human correlate.
Interesting, Troy, thank you. Is there any homology of the dermatopontin to VLA4 or the receptor for VLA4?
Yes. So that area of research we actually just finished collecting all the data on. And so one of the receptors for VLA4 turns out to be VCAM. And so we've actually shown that dermatopontin can actually bind VLA4 on one end, and on the other end it binds VCAM. So it sets up a perfect blockade to inhibit that action. So very intuitive.
Really nice talk, Troy, and terrific work. Do you think that the DPT can play or plays a role at all in the TMA that's seen in patients post-transplant? And, you said that it doesn't matter what the cause of the injury for the endothelial cells is. Have you tried a Shiga toxin? So like looking at the HUS, because that's kind of where the Shiga toxin hones. And so I don't know if it's possible that may play a role in the patients who develop TMA or not.
I think it's a completely wide open question. I think it's very interesting. I don't see why it couldn't play a role. We've not tried Shiga toxin. We've thought about doing it, but we've not tried that. But you're theorizing that like in a small vascular disease setting where you have lots of injury, perhaps DPT is there actually trying to heal the vessels, but also clogging them up. Yeah, no, it's something we could certainly investigate, but it'd be very interesting. We haven't looked at it. Thanks.
You have been disconnected from the meeting.